Hello and welcome to the latest MS Hear from the Experts webinar. This series is helping people better understand multiple sclerosis, highlights MS-related resources, plus provides tools and tips to navigate your MS journey with more knowledge and confidence. Our intention is to help you learn more about this disease, treatments, research, wellness strategies, our programs, and much more. My name is Gabrielle Vito, and I'm a longtime volunteer with MS Canada. My own diagnosis was way back in 1996, and I'm a person living with relapsing remitting multiple sclerosis with the onset of fatigue. It's a real privilege for me to be a volunteer for MS Canada, and I'm very, very happy to be your moderator for today's broadcast. Now, we want to welcome both audiences, including those joining us today from Facebook. Before we get started, we're going to briefly go over a couple of housekeeping items. Your microphones have been muted and will remain muted for the entirety of today's broadcast. If you have a question, please type it into the chat box and we'll try to get to as many questions as possible following today's formal presentation. Now, please note that questions pertaining to specific personal situations likely cannot be answered in this type of a venue, and we recommend that you contact your personal MS help. The MS session is being recorded and will be available soon on our website in English under archived webinars and on the YouTube channel. A Facebook Live recording will also be available. Also, please note, as always, MS Canada does not approve, endorse, or recommend any specific product, therapy, or service, but provides information to assist individuals in making their own health and wellness decisions. Now, today's broadcast is an opportunity to learn more about stem cell research and how it can help to treat MS. Today's expert is well known in the MS community. It is Dr. Mark Friedman. He's currently a professor of medicine in the field of neurology at the University of Ottawa, as well as director of the MS Research Unit at the Ottawa Hospital General Campus, and a senior scientist at the Ottawa Hospital Research Institute. A graduate of the University of Toronto, Dr. Friedman holds his master's degree in molecular neurochemistry and continued his postgraduate work specializing in neurology and neuroimmunology. His specialized training has taken him all over the world to the Weizmann Institute of Science in Israel, the National Hospital Queen Square, London in the UK, as well as the Montreal Neurological Institute, where he subsequently was an assistant professor. He holds his specialist certification in Quebec and all across Canada, and is a fellow of both the American Neurological Association and the American Academy of Neurology. Dr. Friedman has published more than 250 pieces, including articles, books, book chapters, and abstracts, and he's been invited to give hundreds of lectures and presentations nationally and internationally. His extensive research includes the area of molecular neurochemistry, cellular immunology, neuroimmunology, and clinical studies in MS. He is currently holding peer-reviewed and industry-related funding for translational research investigating immune mechanisms of damage in MS, with particular interest in the role of gamma-delta T cells. He's also the lead investigator in the Canadian bone marrow transplant study in MS and co-chair of the International Mesenchymal Stem Cell Transplantation Study Group. Dr. Friedman has more than 25 years of experience in the management of patients with multiple sclerosis and has been the principal investigator on numerous clinical trials with new therapeutic agents for MS. He has experience in serving on several research study steering committees as well as data safety monitoring boards. He serves on editorial boards for several journals, including the Multiple Sclerosis Journal. He's also served on several national and international committees and is currently the treasurer of the America's Committee for Treatment and Research in MS. So we're very thrilled that such an accomplished human is joining us today. Please join me in welcoming Dr. Mark Friedman.
Thank you, Gabrielle. I'm going to have to get the society a little bit of an updated bio. That one's about 10 years old. I'm no longer the treasurer of ACTRIMS. I'm actually the president, but that's okay. But thank you for the kind introduction.
Thank you for letting us know.
I definitely need an updated bio. It's my pleasure to talk to you about a subject very dear to my heart, which is stem cell research and the promise, I think, of being able to harness naturally occurring cells in our biosphere that have the opportunity and capacity to perhaps repair. And this is an evolving field. It continues to evolve weekly, monthly. It's really taking off now that we have the ability to culture cells in a way that are safe, and we can then put them back into people. But the studies are slow in coming, and of course, we are hopeful that eventually they will turn into something really positive for our patients and look to repair. But in the meantime, let me tell you about what has been shown and what we can do today, and review some of the data that we have generated in our own studies here in Canada. So I actually have no disclosures here to tell you about, and here are our objectives tonight. I'm going to discuss stem cells in general, so you have an idea of what we're talking about. I'm really going to focus in on the bone marrow-derived stem cells and how we're going to use them, and importantly, point out the difference between the various procedures that are used in what has been referred to as HSCT. The HSCT is basically hematopoietic stem cell transplants, and they are done in various ways, and I'll go over that for you. And then finally, just summarize a little bit about the mesenchymal stem cell field. Okay, so then let's start with, what is a stem cell? Well, everybody knows that people are offering stem cells, and you can go, and this was supposed to be a nice big build slide for all the places that are advertising stem cell treatments that are going to cure multiple sclerosis today. And my point was none of them actually offer you anything that is of benefit, but more they rob you of significant amounts of money. In fact, when this little study was done a few years ago, they looked at everybody raising money for crowdfunding. And the very top list here, I don't know if you can see it, but there's, I have no idea if you can see my mouse, but it's right at the very top. And there's the number one treatment that people are raising money for traveling to get what people are offering in terms of stem cells. And you can look over here to the right, the various places around the world that are offering these types of treatments with absolutely no guarantees. So that's the MS group. What are stem cells? So I think, the important definition here is that a true stem cell is something that has come from almost as early as the egg that created the human. And this cell is uncommitted. It doesn't necessarily have a role. It could be anything. And it's in such an early stage that it can not only differentiate, which is evolve or morph into a brain cell, a heart cell, or whatever else, but it can self-renew. And so stem cells are really important because when you get them, you know that this is a source whereby you can grow lots of different things. And our ability to identify these cells and culture them outside the body has really led to a greater understanding of what their potential will be. So the hope is that they could be morphed into repair cells, and these cells could then go in and fix damage wherever it is in the body. And that's what everybody's purporting to sell you when you go and get stem cells for various treatments. But the proof is really not there. One of the best things that they've been able to do, and something that we've tried to harness, is their ability to control inflammation. And that is probably the number one reason why we moved to mesenchymal stem cells some years ago. There is also the possibility that if they can get into the right tissue, and so how do you get them in there? Various ways, but let's just say the simplest would be to introduce them intravenously. They will find their way to wherever the damaged tissue is, and once there, they can either promote the local tissue to repair itself, or at least secrete things that allow it to the organ to sustain further damage, so protect it. So that's the hope of this. So different types of cells you can see here, and the different terminologies that are used. Right in the middle here, this is the inner cell mass of an egg, a growing embryo. It doesn't even look like anything yet. This is where we're going to get our embryonic stem cells from. So when I talk about embryonic stem cells, it's taken from the egg at a very early stage before it differentiates into something that looks like this, which kind of looks like it's going to become something, right? There are cord blood cells, that is from the baby and the placenta, and that may be of use for the baby in the future at some point. And then there's adult stem cells that are difficult to get, but from the bone marrow, it's somewhat easier than trying to find them in other organ systems. So the four main sources of neural stem cells would be the embryonic stem cells that I just showed you. Something that's called iPSCs, which are induced pluripotential stem cells, we can make that happen outside the body. And the person who discovered the four factors that you can apply to a cell that will make it go backwards in time and become a dedifferentiated cell won the Nobel Prize some years ago. So this was a very important step in the understanding of stem cells. You can get progenitor cells from the adults, and some cases, at least from the animals, we're able to derive them from the nose, from the olfactory epithelium, or from around the brain itself. And then the mesenchymal stem cells, which can come from various sources, most commonly either the bone marrow, or many people get it from adipose tissue, which is fat. So it's amazing. Some people will offer, or some places will offer you liposuction in the morning and give you your stem cells in the afternoon. And it's a little bit of nonsense, but because the true mesenchymal stem cell needs to be grown. Okay, embryonic stem cells are pluripotent, and they come from that little diagram I showed you, the inner cell mass of the growing egg. And they're really interesting because they continuously self-renew, and they can differentiate into almost any part of the body. So that's really important. But the issue with the embryonic stem cells has been safety, because in the body, they can differentiate into organs, and the body constrains their ability to grow. But in some cases, when you put them in without those controls, they've done some damage. They've grown and they've actually become tumors. So they don't sort of listen to what the body has to say. They're now grown in a unsecured manner. And so people still are quite divided on whether or not we should be using embryos. And I don't want to get into the ethical discussion about this as to at what point the embryo is truly a human and we are destroying humans, etc. So this whole field of embryonic stem cells is still in a bit of a flux, and exactly how we're going to be able to use the cells is still up to some regulators. Although I think it's in Canada a little bit easier than in some countries. Okay, so the induced pluripotential stem cells, as I mentioned, there's four factors here. These factors will literally, you could take a scraping from inside your mouth, an adult cell, fully differentiated cheek cell, apply these four factors, and they go back in time. They literally go back as if they were coming from that egg. And then these are the three major layers of the embryo that then turn into any part of your body. The one we like to talk about a lot is the ectoderm, because it really gives rise to the brain. So you could actually take these cells and turn them into brain cells through iPSC. The problem is one of these factors is c-Myc, and that's known as an oncogene, and that can actually cause cancer. So exactly how we're going to use iPSCs is probably a bit into the future, but you have the potential here of making really almost anything. And as this joke would imply, you could probably go to your local stem cell depot and pick up what you need if we're using iPSCs. They're quite powerful. The neural stem cells you can get from fetal tissue as well, or you can take it from around the brains of mammals, as people have done. And you can grow these into so-called neurospheres. And then these can be differentiated along the line of the three major types of cells you'll find in the brain. The neuron, right? The oligodendrocyte, which is of interest to you all because it's the cell that makes the myelin of the central nervous system that is the oligodendrocyte, and the astrocyte is a support cell. And you can do this using the various factors and grow them. And in fact, if you did that, this group in San Rafael Hospital in Milan has taken this to the next step. And now this is a group who's been trying to do this for 10 years, and they finally got the protocol and were allowed to put it into 12 patients. They grew up neural stem cells from fetal tissue, and they gave it to almost essentially patients who were barely walking at all. They had to be 7 or more on the EDSS scale, which is taking a few steps. And they injected them through a lumbar puncture needle into the nervous system, and they followed those patients for 2 years to make sure that nothing terrible happened. And you can read about it. It just got published in Nature Medicine. There were numerous analyses, and obviously the hope was that these cells would do all the things I told you about, provide support to protect the brain, turn down the inflammation, promote internal repair. That sort of thing is what they were hoping to look for, but what they actually saw was something else. So they took the fetal tissue, they grew it up, and this is in a specialized laboratory called a GMP facility. We have one in Ottawa. They're very few and far between. They're very expensive setups in order to be able to grow the cells according to the standard that Health Canada would insist upon, or whatever the national requirements are. And then through a lumbar puncture needle would introduce it. This is what they're referring to as their medicinal products. And what they did is a whole bunch of tests to make sure that they were safe. And that's key, right? And what they found after all this period of time, they did a bunch of MRIs. They gave another injection actually at week 12. So they gave 2 injections and then they followed the patients. But because these cells are coming from fetal tissue, they don't match the host. And so as a result of that, they had to give drugs like prednisone and tacrolimus, which is an anti-rejection drug, to everyone. Otherwise, their body's own immune system could easily attack and destroy these neural stem cells. I think the really nice thing about it is that they started to look for some outcome measures that are possibly related to protection, like the loss of brain volume with sophisticated MRIs. And they did see a little bit of lower rates of gray matter atrophy. That was the only statistically significant measure here, is this loss of brain atrophy. There were some trends, especially in the patients who had the lowest number of cells versus the highest number of cells. The more cells that were given, they had the better effect on the brain. And there were some trends to having perhaps less progression in the clinical measures, but there's only 12 patients here. So really the trends were there. Actually, one patient did have a relapse with a new spinal cord lesion. 50% of patients continue to develop new lesions. So this is not really a great treatment as an anti-inflammatory, but the measures on the gray atrophy was interesting. There wasn't a change in the biomarkers. And one very interesting finding was they went 3 months later and retapped the patients for spinal fluid, and they were able to find, at least in one patient, evidence that those cells survived and were still there. So promising, very early, very, very early study of neural stem cells. And so obviously that warrants a little bit more of study. Okay, in the bone marrow, we have the cells that make up your blood, your red white cells, but you also have these so-called stromal cells, or support cells, that create the scaffolding where all this grows. And it turns out amongst the scaffolding cells are actually cells that have interesting properties, and they are the mesenchymal stem cells. And you can grow these very easily in a test tube and purify them down. And they have all sorts of very interesting qualities, but one of the ones that, and this is just this is how you characterize them, but they can form all the different layers of the embryo. So these cells can actually be quite pluripotent, or multipotent as it is here. They have some distinctive characteristics, and this is why we wanted to use them for something like MS. There is some thought that they could turn into the cells like the oligodendrocyte, but there's very little proof of that. And there is some thought that because they can release all these factors locally, they might actually promote repair. But probably the best evidence is that they are anti-inflammatory. This is not what we need today. We have plenty of drugs that are anti-inflammatory. We were hoping to be able to show with mesenchymal stem cells some repair. And these are all the different things that the mesenchymal stem cells can do. So animal models have showed that they work on remyelination, reduce scarring, help blood supply, suppress the inflammation, can be shown to have neuroprotection, and even help cells by becoming what we call a cell fusion product with them. All very interesting. All of this shown in animals. None of it has been proven in humans. However, there are a number of different trials that are out there trying to show that mesenchymal stem cells might do something, and I'll touch on a couple of the major ones. But if you have aggressive MS, I mean, really, the game changer here has been the ability to knock out all this inflammation, and that's what we've done in our pioneered studies here in Ottawa, where we use the hematopoietic stem cells to support and regrow an immune system in patients who we've been able to eliminate the disease-causing immune system. Without the ability to renew the entire hematopoietic stem cell system, that's the red and white cells, we wouldn't be able to remove the old one. And that's really where the stem cells have helped us. So I'm hoping this slide tells you the difference between these two. What we were calling bone marrow transplants, or HSCT, is removing the old or diseased immune system, and that is usually done today with chemotherapy. And then we grow a brand new immune system, and we do that from bone marrow derived stem cells from the same person, so that you don't have rejection as a possibility at all. So that's the bone marrow transplant, HSCT. MSCT is the mesenchymal stem cells. You get them from the bone marrow, you grow them up. It takes about a month in a facility, and then you can inject them back even intravenously. In the case of MSCT, there is no use of chemotherapy. So none of the problems that are associated with chemotherapy are you going to find when you use mesenchymal stem cells. Some groups have claimed to be able to see something with IV. I'll show you a group that's using a mixture of intravenous and intrathecal. That means introduced through the spinal cord lumbar puncture needle. Okay, now the hematopoietic system, that's different, right? This is the stem cells that we're going to use to replenish the immune system in somebody in who we've eliminated it. And with a single stem cells that we can actually derive, and you don't even have to take them out of the bone marrow now, you can chase them out of the bone marrow with some drugs and then collect them on a machine I'll show you in a minute. Those cells can give rise to everything you see here, which is part of the hematopoietic stem cell system. And in addition, it will also become your very important immune system. So the T and B cells you all hear about, they are all derived from the same stem cell. When we started this project to replace the immune system at the start of the last millennium, the theory was that if we couldn't figure out what the immune error was in any one patient, it seemed to be different in everybody. What if we just completely removed the old immune system and introduced a new one from their own stem cells? And theoretically, if it grew up, and it grew up in the environment of disease with the myelin pieces all rolling around, they'll just grow up in that atmosphere and think that those whatever myelin is part of the body, and therefore won't reject it. So it's called induced tolerance. We knew that we could do this, but then there was some data that was actually showing that these stem cells that we were going to use to renew the immune system are actually stem cells that could do a lot of things. And maybe that's part of why we were seeing so much recovery. But I think you've seen all the data from our HSCT trials. Now, the reason why we moved on this is because some patients who had bone marrow transplants because they had leukemia survived their leukemia, but their MS got better. And so we went, what's going on? Obviously, this bone marrow transplant did more for the MS as well as whatever the malignancy was. So these were the observational studies that sort of led to the very first trials. There are two types of protocols that are used for HSCT. The immunoablative protocols, which I call chemo light, because They don't completely remove the old immune system, and therefore they fail. Or the fully myeloablative, complete removal of the old immune system, and because we don't know who the bad guy really is, but if we can remove all those cells, then and grow a brand new one, then maybe it won't attack anymore. And those are the myeloablative protocols that is similar to what we've used. So myeloablative is probably the more horrendous of the two, and we use very high doses of these drugs that are listed here, whereas the lymphoablative tends to spare the bone marrow, and that means less morbidity to the patient, less chance of dying, but also a less chance of controlling the disease. And the most popular amongst these is this so-called BEAM, which stands for all these drugs over here. That's used pretty well in Europe and many sites in the US because it's less complicated. It tends to produce an effect, but it's not 100%, as you'll see. Okay? So where would we use this? Well, we have many potent medicines today for controlling inflammation, but even in some patients, those medicines are not enough, and we know that. And so if you have a very resistant form of early disease, the idea is not to go shopping through all the different drugs that we have today and fail in every one of them. But if it's recognized that you have one of these aggressive forms of MS, then the patients are often referred for a bone marrow transplant. And right now, other than Ottawa, the only other center I know that's offering it, and they've just started, was Calgary. So the immunoablative protocols have less morbidity, means you don't get as sick, but they also produce less than 100% disease control with return of disease activity within 2 years in about 20 to 25% of patients. So if you're going to go through this whole procedure, I would think you'd want to do better than just 2 years, because we can get you 2 years just with some of our medicines. So when we launched this project was to see whether or not we could get a much longer, a durable response in patients without having to retreat them. And that's what we did. And this is how we did it. And this is all more or less published. But there are 4 different phases, and all the little things that go in here are very jargony, so I'm not going to go into it. But you have to mobilize the stem cells. You have to collect them and prepare them. And we do this extra step to prepare them, which has actually 2 objectives. Not only does it purify the stem cells, but by so purifying the stem cells, we remove the potential cells that might also recause the disease. So we're cleaning the graft, more or less. And then you go through this period of time where we remove the old immune system. That's called conditioning. And then the most anticlimactic part is when we just thaw this prepared stem cell collection that's kept for us at Canadian Blood Services. We literally thaw the bag at the bedside, let it drip into the patients, give them something called filgrastim to help them engraft, and give them a little bit of rabbit antithymocyte globulin just in case one of the bad guys crept into that graft. And then we wait for the engraftment. So this is the technique that we use to purify the CD34 cells. This is done all by Canadian Blood Services. This is a leukapheresis machine, literally one vein in one needle in this arm and another needle in the other arm. The blood passes through, the white cells are collected and come back into the other arm. That's how we get them. And then we do this immunomagnetic separation for a marker called CD34. And anything that's not CD34 gets washed through. So that at the end here, you see all these cells over here. There are probably some of the bad guys in there. We toss them. And then we release the magnet and collect all the CD34 cells, and you've got a purified graft. These were the patients that we did. This was all published in the Lancet about 6 years ago. So then about half the patients were already in a progressive phase, and many of them were at the borderline of not walking. So we did 24 patients, and we did 8 controls. And we've done lots more. This is an old slide. We're probably over 100 patients now that we've done for Canada. And our results, I think you're all aware of this, even after 23 years, there wasn't a single new attack in anybody. There wasn't a single new MRI lesion in anybody. And the surprise was that some of the patients who had already reached the stage of walking with a cane started to get better, and many of them actually improved, and that was totally unexpected. So repair is definitely possible, and that was one of the lessons we learned from this. And whether the stem cells we were putting in to regrow the immune system were part of that repair, that seemed to be the focus of our next grant that we got from the MS Society, to study what qualities of those cells might confer repair. And I wish I could tell you we found something, but we haven't been able to find anything about those cells that made it a difference for the patients getting better or not. We just know that some of them got better and others didn't. So you can see here, no relapses after the HSCT, and the relapse rate was effectively 0. And when you look at EDSS, and you're all aware of this heat map, which I always apologize because it was made by my colleague, Dr. Atkins. Normally you think of red as bad and green as good, but to a hematologist, red is good. So he's made them backwards. And you can see here that there are a lot of reds here on the EDSS. So these are patients who've actually improved their EDSS. Most of the patients either stabilized or improved. There was a small subset that actually got worse. And again, we can't tell you which of the patients at the beginning were the ones who are going to get worse and the ones who are going to get better. We know that the longer you've had MS and the more damage that has occurred, it's harder to repair. And some of the things that we looked at were, I think, quite significant. When you look at the patients who progressed versus stable or improved, things like being able to work, going on long-term disability, whether their relationships survive, whether they can still drive. And you can see there's very little in the patients who progressed, but in the patients who actually got better, and that's the majority, you had an increase in the people who actually returned to work, got off of long-term disability, preserved their relationships, and even got back to driving a motor vehicle. From the MRI standpoint, literally they had so much activity at the beginning, and then right after the transplant, 0. Basically, no new MRI lesions. One of the most fascinating things was, as I talked to you about the neural stem cells, and one of the outcome measures was this loss of brain tissue, that it reduced the loss of brain tissue. We saw this in our MS patients here. So if you ignore the first year here, this line, I'm hoping you can see the marker, but this gray sort of area here is how much brain you would lose if you had no treatment at all, if you're an MS patient. And the big solid line up here is actually how much brain a normal person would lose for the age. And after 12 to 24 months, it was a little discouraging because the rate of brain loss was the same as if we didn't do the transplant at all. But if you wait, that's where we started to see it, because in years 3, 4, and 5, the rate of brain loss just stopped. And by the end of 5 years, these patients are losing brain at the same rate as an age-matched control, as if they didn't have MS. So there are some things that are reversible. This is just a cartoon showing you all the relapses before and none of them after. And then when you look at the MRI had lots of damage before, and 0 afterwards. This is just the clinical. Here's the MRI, no new enhancing or new lesions. And these are all the MRIs everybody got after the transplant. So quite a few looks, but over the last 20 years, we've not seen anybody recur. So in summary, this is a way of stopping all the inflammation in somebody who has a very aggressive form of MS. The promotion of recovery is something that we're still trying to understand. And what I didn't mention to you, and obviously the reason why our government wants to pay for this, is nobody has needed to go on a disease-modifying medication ever again. So that's a huge savings to our healthcare system. This is a quick comparison of the 2 other major studies that did bone marrow transplants differently than us. These are the Americans in the HALT-MS study. Again, they used an immunoablative protocol, BEAM-ATG. This is Richard Burt's stuff in Chicago, where he used an even lighter form of immunoablation, so just a low dose of Cytoxan. And this is us. And so this is the probability of disease progression, which occurred, if you look after the first 2 years, nothing. Whereas if you look at these other two studies, there are people falling off the curve as you go out here to the number of months. So as they fall off the curve, they developed new problems, new relapses, new progression, or new MRI activity. So we're the only ones who didn't have that at all. When we look at so-called NEDA, which is no evidence of disease activity, we're over here. Where is it? Right there. And so we were able to sustain really no evidence of disease activity for much longer than other patients. So the Canadian protocol is probably the most aggressive one. This is lymphoablation versus myeloablation. You can see we're way up at the top here. We're the most aggressive protocol, but we also have the best results compared to any of the other groups. We've learned that age is important. As you get older, you're less likely to repair. And so, it's not surprisingly, the older patients here didn't do as well. This is progression-free survival, and they're way down here versus the younger patients over here. And if you're still in the relapsing phase, you do a lot better than if you're already in one of these progressive phases where there really isn't that much inflammation to target. So pretty well, if you're non-active secondary progressive or primary progressive MS, you're not going to benefit from a bone marrow transplant or HSCT. This is a very difficult, a little bit to understand graph here, but I'm just going to point out, this is the Canadian study here. This is a 100% line here. So that is 100% relapse-free, more compared to the other groups here, where after 2 years, they start to have return of disease activity and then often have to go on to medicine. So we're the only study 100% relapse-free. If you want to compare how bone marrow transplant does to all the other drugs that we have today, this is all the other drugs we have today. And you'll notice they're all things are to the right here, meaning they all start to have relapses again. There's only one that has no relapse, that's us. The others have also had this. And so what we did 20 years ago was to get a study through the MS Society's coverage for the treatment. And so we were very limited, but what we're doing today is a little different. We are still taking younger patients. We're looking for the early aggression. This is characterized by lots of attacks that leave you with some residual deficits, despite usually an attempt at therapy, and an early progression with usually a big burden on MRI. And obviously, no other significant risks because you're enduring a very horrendous procedure. So I think in conclusion, most people have agreed today, and I just saw another paper come out from something like 135 transplants they've done in Sweden. They just released that data today, and not surprisingly, they're seeing the same sort of phenomenon, that if you do it, most of these patients do extremely well for a long period of time, do not need any kind of disease-modifying medication. So patient selection, very important. The type of conditioning regimen is key, because if you're only immunoablative, there's a good chance the disease is going to come back. The delayed recovery is very fascinating, and we're still trying to understand why that occurs and why some people do so much better than others. And that's really a focus of continued research. All right, so I promised you to hear something about the mesenchymal stem cells. These are the support cells from the bone marrow. We can get them the same way we can get the other stem cells for bone marrow transplant, but they're a little more tedious. So we actually have to go into the bone marrow and extract the fluid, and then we grow it in a specialized facility. And this is what it looks like. This is not your fly-by-night centers who take your adipose tissue or fat tissue in the morning and liposuction and give you back stem cells later in the afternoon. No. This stuff has to be grown in a very, very specialized facility that is approved by Health Canada, so-called good manufacturing process. And they're grown to large numbers so that we can give injections of 2 or 3 million cells per kilo. That's what you're looking at, infusing into people. And so MESCAMS was the Canadian arm of the MESMS project, which is the international study I organized together with Antonio Uccelli around the world. You can see here all the different places that were part of this mesenchymal stem cell trial that we did, and we got funding from everybody. The Canadian group was actually funded by a combination of the MS Society of Canada and Research Manitoba, because it was also done in Winnipeg. And so it was a very interesting design. Everybody got cells. They either got them at the beginning or after 6 months. And we followed them meticulously for any change clinically and by MRI. And the aim was to show that if we injected these, they were safe, they didn't cause any problems. And that was number one aim. And then we were looking at, hopefully, a biological measure, which is to reduce some of the inflammation, which we know they are capable of doing, at least in mice and in some of the human projects. So this is just a breakdown of how everybody got screened and eventually ended up in one arm or the other. But after 6 months, if you got the sham cells at the beginning, you got the other cells. But our readout was really at 6 months. And so these are the different groups here. There's the placebo. You see the placebo moved over and got the cells, and vice versa. And then we looked at some outcomes. And I wish I could tell you something that it really showed us benefit. These were trends. Okay, so the blue is the people who got it at the beginning. The red is the people who got it after 6 months. It looks like the number of enhancing lesions went down, but none of these measures were actually statistically significant. So there was just a very active group of patients. Actually, we had many attacks during this study. People were very, very active. You can see here the relapse rate looks like it's trending lower compared to placebo, but again, statistics were against us here, and we didn't see this as a significant outcome. So we know they're safe, and given intravenously, they didn't have the effect on the MRI measure that we're looking at. Therefore, we had no way of reading out anything else, because that was our control. Like anybody who had a reduction in the MRI, we could say, okay, they had a biological effect. Let's see what else it did. And we've been trying to study the analysis to see if there are some people who did better than others. Now, another group in Israel showed that they did get some effect. But when you look at the study, it's the numbers pared down to such small numbers, it was really hard to make some understanding of it. But they did very similar. They took the mesenchymal stem cells, they grew it up in a proper facility, they tested them all, they gave 1 million cells per kilogram body weight, but they divided it. They took 48 patients and they divided this now into 3. They either got nothing, or they got an IV, or they got an intrathecal, meaning through the lumbar puncture needle. And then they broke that into a second cycle where either you got the mesenchymals in the spinal cord again and nothing else, or some of them got the IV and nothing else, and some of them got both. And then they looked at some of the outcomes. And this has all been published. Same type of outcome measures, safety, and did it do anything to the usual things that we measure in patients over time. Now, unlike our group, they actually started to see some differences that were clinically or statistically significant. This is who they treated. So they gave PPMS and secondary progressive MS patients. They took a whole bunch of different types of MS and treated it. I don't want to get into the details here, but there were really no adverse events except headache. And the reason for the headache is in those patients who got the lumbar puncture needle, you can get a headache afterwards. So they looked at these 2 cycles, and this is sort of the run-in period and the different cycles that they give post. And then they looked at the change in EDSS. So in their case, their EDSS actually improved, especially with the intrathecal, the cells that were given into the spinal cord. This is a little bit more hard to understand, but the number of relapses that were occurring here, you can see here the placebo group versus the intravenous mesenchymal stem cells versus the intrathecal. So the intrathecal actually had almost no relapses. So it looked like sending the cells directly into the nervous system made a difference. And this is the NIDA. That's no evidence of disease activity. That's no relapse, no new MRI lesion, or no progression. They got that in about 58% of the patients. No real placebo control, so you don't know how they would have done on top of that, but it was statistically significant for the intrathecal group. And they continue to do intrathecal as well as intravenous. But they believe that the intrathecal route is probably the way to go, and that's in their outcome measure. We did not do the intrathecal route. We went intravenous. Some people believe strongly that the intravenous is still important because the first place it will go when you inject it intravenously is the lung. And the lung turns out to be a very important immune organ. We discovered it the hard way the last few years. COVID taught us a lot of lessons about how much immune function comes from the lung. And these cells actually get processed in the lung. And when you do tracer type of analyses in mice, even though you give it intravenously, you could look 2 weeks later and they're in the brain. So we know that the lung does process them in a way that perhaps makes these cells able to do the things that they're able to do. So I think that these studies are suggestive, but they need more definitive proof. And I know these other studies are actually being planned. My advice is to stay away from these medical tourisms that are looking to take your money and take desperate people and make them no good promises. But that's just my own feeling about that. I think we need to see the proof before we would go ahead and consider the treatments. So, I mean, obviously a lot of people to thank, including the MS Society of Canada, who's helping to put this on tonight, but all the different people that were involved in these studies are probably too numerous to mention here. And I think I'm stopping in enough time to allow some questions. Laurie, I promised you 5 or 10 minutes, so here you go.
That's fabulous, Dr. Freeman. Thank you very, very much. We are listening to Dr. Mark Freedman today. He is joining us from Ottawa. He is currently the president of ACTRIMS, the America's Committee for Treatment and Research in MS, as well as the director of the Multiple Sclerosis Research Unit at the University of Ottawa. So thank you for being here. We do have some questions coming in, so I'm going to jump right into them. You did touch on this first question in one of your slides, talking about the importance of patient selection. This person is asking, I have primary progressive MS. Would I benefit from stem cell therapy? I was diagnosed in March of 2022, so fairly recently.
So the primary progressive, secondary progressive thing is changing dramatically. We just think of MS as probably progressive from the outset, and in some people we see relapses. But the relapses and the new MRI lesions are a reflection of the type of inflammation that can be treated with HSCT or the bone marrow transplants. When you don't have that anymore, then something like a bone marrow transplant is probably not going to benefit you. So will it work in primary progressive? I'm not lying. We've done a couple of primary progressive patients, but they were very inflammatory. So they are people who progressed, but at the same time they're progressing, their brains were lighting up with new lesions, and they actually got attacks. Just because you have primary progressive doesn't mean you're not allowed to have attack. In fact, all the studies that have looked at primary progressive MS, 20 to 25% of the patients start having attacks. So if you still have that inflammatory phase, there's no reason why it would not work.
Excellent, thank you. Moving along, I'm going to go to a question that is coming from the parents of a daughter who it sounds like has MS. So they're saying, our daughter's pregnant and due in November. We've discussed preserving the umbilical cord. Where would we go to find additional information, the cost for preservation, and potential use for MS-related stem cell transplant?
And I get this question a lot from patients because they think that they're able to harness the cells of their daughters, sons, whoever. I mean, babies are coming, so you can use these cells. They are not a match. They're not even a match for the mother. The only person those cells are going to be good for are the baby. So I would say you want to preserve the cells. There's lots of places who will do it. There's the cost of the initial extraction, which is not cheap. And then there's almost like a monthly fee to keep the cells frozen, and because that requires some maintenance. But those cells are not going to benefit mom or anyone else. The only person they're going to benefit is potentially the child. Should that child grow up and have something that might be needing the stem cells, then they would be able to use it, but no one else can. because otherwise they're not a match.
Excellent information. Thank you. Okay, I'm going to move along now to a person asking about comparisons of data in data. What is the comparison data of MST and HSCT in regards to success rates?
Well, I hope that person was listening, because HSCT and MSCT are completely different. HSCT is the removal of the old immune system and replacing it with a new one. And the various groups that have done that have seen tremendous success at stopping new activity. The ones that use the myeloablative procedure like we do have eliminated all the activity. Others eliminated most, but not all. And there's a disease coming back in the way of relapses and new MRI lesions in about 2 to 3 years. MSCT at this stage is unproven. We know what the potential is, but there's small studies. So far, they're safe, but they have not shown to do anything for the disease. I hope I've answered that question.
Yes, I think you have very clearly. Now, this next question is someone asking about a specific treatment that's on the market, Mavenclad. They're asking AHSCT sounds like an aggressive version of Mavenclad, as Mavenclad uses a type of chemo to lower the immune system, and the hope is when it regenerates, it comes back less aggressive. How much riskier is AHSCT in comparison?
Well, that person is absolutely correct. Cladribine, which is Mavenclad, has been used by hematologists for years to reduce the cells of the immune system, especially if you have a leukemia, and there's a certain type of leukemia they've been using. We've been using it for years for MS until the oral came out, and then we started using it orally. The difference is if it's to make the comparison, what I was talking about earlier, cladribine is at most immunoablative. So it's not completely fixing the problem. Some patients have done amazingly well on the cladribine and have not needed to go beyond to HSCT. We've had others who broke through the cladribine, and we did do HSCT. There was another drug similar to cladribine, like what's not used very much today, is also alemtuzumab. And that was a drug that also the hematologists used to prepare the bone marrow for actually HSCT. So they're all in the same sphere, but HSCT is the complete removal of the immune system. And when you do that, you put patients at a much higher risk of having problems. It turns out the biggest risk has been the toxicity of the chemo drugs themselves, and less so the openness to infection. So cladribine is a light version of HSCT. If cladribine is here, HSCT is way up here in terms of its strength and its potential for causing problems.
Excellent information. Okay, thank you, Dr. Freeman. Okay, here is someone who has a bit of a multi-section question, but I think they're all very relevant to what we've been talking about. They're saying amazing findings. Now, how can a patient be qualified for this treatment, and why aren't relapsing remitting patients considered if it's better to get the treatment early? I was planning to go to Mexico. So here's someone who is considering that medical tourism that you were talking about.
Yeah, absolutely, it's for relapsing remitting. It actually, I'm trying to get rid of this terminology and just talk about MS as an entirety, and that's what the world is focused on this. There was a consensus paper that Tanya Kuhlman released last year. I'm sure you're well aware of that, Gabrielle, where they want to just look at MS as a spectrum from start to finish. It's probably a progressive disease to start. You don't see the progression because it's masked by all the relapses in people. But eventually, when the relapses stop, you can see that there was some progression underneath it all. So it's for anyone who has the focus, which is that inflammation. As long as you have that inflammation, call MS whatever you want. Relapsing, secondary, progressive, primary, doesn't matter to me. If it's early, if it's progressing with this inflammation, and it's not being taken care of by one or two trials of the medicines we have today, then there's no reason why we couldn't see those patients and consider them for HSCT. What they're doing down in Mexico, I can tell you, they're using chemo light, so they're not going heavy duty chemo. You don't even need the stem cells, but they do it because that's where they get the money, 65,000 US dollars. And then they know damn well that's not going to work, so they put everybody on rituximab, which is a generic version of two of the very successful anti-CD20 therapies we have today, ofatumumab and ocrelizumab. But rituximab is not licensed in Ontario it's actually not licensed anywhere in Canada for the use of for treating MS. Unfortunately, in British Columbia, they don't want to pay for the other drugs, so they actually, if you wrote a prescription for ocrelizumab, you'd get rituximab in BC. But that's what they're doing in Mexico. They're basically, you're spending 65 grand for generic rituximab. That's basically what you're getting.
Also, excellent information. I'm so glad we have you here tonight. Okay, I'm going to, in the interest of time, combine a couple of questions that have come in, and you've touched on it already. The differences across Canada already exist in MS treatment. Do you see, on a go-forward basis, any wider availability of what you're doing in Ottawa to MS patients across Canada?
Lord knows we've tried, Gabriella. We've tried to get other groups interested. I had a fellow just spend 2 years with me, and he's gone back to Quebec City, and he's pretty sure they're going to be able to start doing transplants there in the fall. There's many reasons why this is not available, and probably the biggest one is lack of resources. There has to be a commitment from whoever's paying the bills to allocate the resources to a bone marrow transplant. We have that in Ottawa. We battled for it in Ontario, and we got it. And the hospital literally put aside these beds with the support from the ministry for what is called non-malignant hematology, because that's the problem. The hematologists are up to their ears in patients with lymphoma, leukemia, myeloma, and they're trying to treat all these very aggressive malignant killers. They don't have the time or efforts to do non-malignant hematology. We were committed along that route here. We got the resources, and we've been fortunate to be able to accommodate Canadians wherever they are in this country, and they've come to us from everywhere. We can do that in Ottawa. It would be lovely to be able to do it elsewhere. And finally, my understanding is Jody Burton out in Calgary has got a group that has done the transplants. We had a patient supposed to come to us from Vancouver, and they canceled their appointment to say they're going to do it in Calgary. But they're not using the same protocol, just so you know. I see all these questions in the chat, but so I don't know if you're looking there or I'm looking, but there's some questions here.
Yeah. I've combined a few of them. The other ones, we're right out of time at this point already. But the other questions are related. Should AHSCT be the first line treatment for MS? People are asking.
Let me answer that.
Okay.
Yes, let me answer that, because in some cases, absolutely. And together with our colleagues around the world, we published that just a couple of years ago, where we used it first line. And it's just, not everybody presents with the same type of MS. And some people come in roaring with disease, unfortunately, and it looks like they've had so much already damage that the bottom line is we got one shot at this. And if you only have one shot at it, what are you going to do? And in that circumstance, we would do HSCT. Keep in mind, 2/3 of the patients are female, and often they're young, 20s and 30s. If you do HSCT the way we're doing it, 100% of the patients are sterile and will not be able to have children by the natural means. We've had several be able to have babies You got to borrow an egg from someone and you fertilize it and you can put it inside. We can do all that today, but it won't be the natural way. So that's why there's a little hesitation here to not just give it to everybody, because you're destroying your fertility. And I think people still want to have a chance at having families. And, but if the disease becomes more aggressive, we can always do that.
Dr. Friedman, I want to thank you so much for your time today. This has been excellent information. You've automatically answered a couple of other questions that we had on the list. We don't have time to get to them all. I'm afraid we're going to have to leave a couple of them there. But thank you so much for joining us. We really, really appreciate you sharing your expertise on this amazing topic to the MS community. So thank you so much.
My pleasure, Gabrielle. Good night, everyone.
All right, everybody, as we wrap up today's broadcast, we want to remind you about the MS Knowledge Network quickly. It's staffed by trained navigators who provide consistent quality information and support. You can connect with them by phone, email, or live chat through the MS Society's website. All interactions are confidential, and the information is trustworthy. MS Navigators can help you learn more about programming and resources that we offer from the Society. Plus, they can help you find other community or government supports and programs. And we can get you signed up for the monthly MS Canada e-newsletter that covers all sorts of information. Now, we invite you to be a part of the MS community and encourage you to get involved with our programs, our services, our events, our government relations, and our volunteer opportunities like the one I'm doing right now. These days, connecting with each other is more important than ever, and all the information on how to volunteer is available on the website, which is now mscanada.ca. Now, if you have questions or need support about this webinar or any of our educational activities, you can reach out to the education team. They have a direct email. It's education@mscanada.ca. On behalf of MS Canada, I want to really thank you for tuning in to today's webinar. Take good care. Bye-bye.